Okay, so my name's Richard Wilson, and I manage the high-resolution mass spectrometer that we have at CSL, and it has a number of applications, but the main one is for proteomics. As James has touched on previously, we also do metabolomic studies as well using this instrument. And for those of you who aren't familiar with proteomics, it's really a term used to describe protein discovery using unbiased methods, and it can range in complexity from quite targeted analysis, where you mainly have a handful of proteins, through to just a fraction from a cellular lysate, through to a whole tissue or organism. And whatever your application, really mass spectrometry is the core technology. As far as this department goes, we've been working with Tia and plant science for a number of years, particularly in microbiology. So the organisms that we look at range from bacteria through to plants and also animal tissues as well. So really covering the whole spectrum of different types of protein samples. So whatever the source of your protein sample, you need an extraction method, and in some cases you may wish to sub-fractionate your proteins. That's sort of a wet lab part of it, and then my role in the process is to analyse the peptides that are produced from trypsin digestion of your proteins using the Orbitrap Tandem mass spectrometer. And then there's the computational aspect, where the acquired mass spectral data is used to interrogate sequence databases for protein identification. So the extraction methods that you use have to be tailored to the type of sample or tissue you're working with. These can be a combination of chemical, enzymatic and mechanical means. You can be selective to enrich a subpopulation or a subproteome of interest, but the more elaborate your protocols are, the more sources of variation there are. So they have to be robust methods. And the methods that you use may well differ from targeted analysis. So you might read a paper that says you can prepare proteins from this particular tissue for a Western blot, but this may involve chemicals such as mass spec unfriendly detergents or exogenous proteins. So you may have to use different methods to targeted analysis in proteomics. And it's always very useful to determine how your extraction procedure, how efficient it is and how reproducible it is using an orthogonal method. So protein assays, as we found out in recent years, can give quite unreliable results, and it's useful to use an alternative method. So up until about a decade ago, the traditional method for proteomics was to fractionate all your proteins using orthogonal methods such as 2D electrophoresis. So here, just to illustrate, we've got 3 replicates of 3 different experimental conditions, control and 2 treatments. Using 2D electrophoresis, you can identify protein differences. So here I've highlighted these proteins which are present in the controls and in this treatment set, but are depleted in this treatment set. So here the emphasis is on protein separation. And then these spots can be excised and then analysed and hopefully identified using mass spectrometry. In recent times, the emphasis has shifted from protein fractionation to peptide fractionation. So in this case, you take a protein sample, digest with usually trypsin is the enzyme of choice, into short peptides, and then we separate these peptides using HPLC. Now in this case, we use analytical columns that are basically packed capillaries, and we use very, very low amounts, separated at very, very low flow rates, and this is by miniaturizing the whole system, it increases the sensitivity, so we can detect many hundreds, if not thousands, of peptides during a single run. And this is what's known as the base peak chromatogram, which represents the most abundant peptides in the mass spec at any given time. So at any given moment during a 2-hour separation, for example, we're interfacing the procedure with the mass spectrometer, and so we're detecting not just a single peptide ion, but also many others as well. And these are targeted for identification. Now, we can use the peptide ion intensity as a method for relative quantitation. So here, for example, I've just shown the extracted chromatogram for one particular peptide in sample A that has an intensity of around 6.3 times 10 to the 7, and sample B it has a much lower intensity, and we can integrate the area under the curve in order to express that data as relative abundance. Another approach to use, which is the one that we use at CSL, is to isolate the ions, the peptides, and fragment them to generate an MS2 spectrum. So we're simultaneously detecting the intact peptides, but we're using a second mass spectrometer to do the fragmentation of those peptides, and that's why this procedure is known as Tandem mass spectrometry. So this just illustrates the fragment ion spectrum for that specific peptide that I've shown here. And this is a useful approach because not only do you quantify these peptides based on the number of times these fragment ion spectra are detected, but you can quantitate their abundance based on those counts, peptide counts. So, as I've shown you, the process involves taking the protein sample, digesting into peptides, and deriving the MS spectra. This is a summary of quite a lot of work, but basically the computational side requires that you have a protein database for the organism you're working with. This is digested in silico to generate a list of predicted peptides, and the peptide mass that's recorded in the mass spectrometer is used to generate a filtered list of possible candidates, and then the MS spectra is used to identify the most likely peptide based on the similarity between the observed MS spectrum and the predicted one. So when you do this process on a complex sample, over 2 hours you can record up to 15,000 MS spectra, and these can match to up to 10,000 sequences in the database, which may correspond to, for example, 1,500 distinct peptide sequences, and then these peptide sequences have to be matched to the proteins, and that's the process by which they're identified and quantified. And it's important to emphasise that this is not an absolute, but it's based on probabilities, and so you can set certainty cutoffs for each of these steps. And depending on whether you want a low false positive rate or you want to basically discover everything that's in the sample, you can tailor these cutoffs to your requirements. And that's just a review that talks in detail about this process. So when we've completed the database searches, the peptide data is uploaded to the server, a lab key server, and this just shows in one particular sample a list of proteins that were identified, and it's been ranked according to the number of peptide spectrum matches. And of course, once you've identified the proteins in one particular sample, the idea is to compare it to others, and so there are a number of statistical methods you can use for count-based data that take into account the consistency between replicates and also the absolute values. So you get more robust measurements when you have more counts being recorded. And this just shows a sort of a null plot where two replicates for the controls have been compared, and then this shows the protein data for one control sample versus a treatment sample. And so you may wish to target the most differentially abundant proteins for subsequent analysis by orthogonal techniques. And that's all I'm going to say, given the amount of time we've got today. Thanks.